Peppers, right? South American cuisine is very spicy, especially when it involves malagueta, which is like the pepper in Brazil. And it's called a malagueta. Yeah, malagueta. Really? What? That's like, oh, is your family from Minas Gerais? They're not. Yeah, malagueta is really huge, especially in Minas Gerais. Yeah, so you find this type of pepper even throughout the West Indies. Like when I was going to med school in Dominica, when I was living in the capital city of Roseau, my landlady in her backyard, she had tons of different fruits and vegetables and stuff. She also had a malagueta tree, which was really cool. So I used to pick my own peppers and prepare them myself. But the reason why I have the opening here is because capsaicin is going to be very similar to heat in that it binds to the TRPV1 receptor. And that's TRP is transient receptor potential type channel, and peppers and heat operate on the same exact type of channel. And it's going to be a calcium channel. So whenever those channels get stimulated, you get an influx of calcium. And we're going to talk about how capsaicin can also be used as a medicine to treat neuropathic pain.
Is that the same as capsicum?
Capsicum? I never heard of capsicum. What's capsicum? Capsaicin is the active ingredient in chili peppers. So like jalapenos, habaneros, all those really intense ones like the Carolina Reaper, all those peppers, capsaicin is going to be the active ingredient. And they actually do sell just pure capsaicin. It doesn't taste great, right? Because you get the entourage effect with peppers. All different peppers have different flavors because it's not just the capsaicin, right? The capsaicin is just the heat element. But again, they operate very similar to just heat. That's why when you have peppers, you feel really warm.
From the sun heat, like that kind of
like heat, just any sort of heat, like fire, flame, sun, all of that. So it's going to be similar type of sensation. That's why it hurts when you have really, really hot, spicy food, and that's why it's awesome. Also, fun thing to, just a side note, chili peppers originated from the Americas. So back in the day, before chili peppers were introduced to places like East Asia, India, the food, well, maybe not so much India because India has lots of different spices as well in general, not just like chili peppers. But in China, Chinese food back in the day was actually pretty bland. It was all just kind of starchy until chili peppers were brought from the Americas to different parts of the world. So, for example, like Szechuan peppers, those are not from Szechuan. Those are not Chinese. Those are actually they're actually derivatives of peppers you find in the Americas, Central America, South America, etc. And then you also find these types of peppers more so in areas that are actually closer to the equator. So equatorial areas that have hotter climates, you'll see more chili pepper use. And that's because it can help to basically prevent spoilage. It can actually help to stave away like parasites and things like that. So super cool. Huge fan of chilies, especially malagueta. So we'll talk about capsaicin and its therapeutic benefits in a little bit. So here's an overview of what we're going to talk about today. It's kind of piggybacking off of part 2 that we were discussing in terms of like ion channels and how they work, electrical conductance. We talked about last time the voltage clamp method. Today we're going to talk about the patch clamp method a little bit. Kind of similar to the voltage clamp method, except with the patch clamp method, you're isolating one individual ion channel, and you're seeing how that one single ion channel is able to actually function. Then we're going to talk about the different types of channels and how they work. So we're going to discuss like the actual the physical nature of voltage-gated channels. Why do they open up when you have depolarization? So it has to do with charged proteins within those channels that are going to be repelled by a positive charge. And so that's why they actually change their conformation, change their shape, and open up, allowing for ions to move through. Then we're going to talk about ligand-gated channels, neurotransmitter versus cyclic nucleotide-gated. Cyclic nucleotide-gated channels basically operate on a second messenger system that involves like cyclic GMP, cyclic AMP, right? So that's why we call it cyclic nucleotide-gated. And then we're going to talk about those transient receptor potential channels, TRP, thermosensitive, including responding to capsaicin, for example, as well as mechanosensitive, which change to touch, for example. So channels that are going to be responsible for the sensation of touch, right, those are going to be involving ion channels that open up just due to like mechanical manipulation of the channel. That's basically a summary of all this, all that we're going to be covering today. So these are the things that affect ion channels. So if you have a large ion current, right, so that's the channels are able to allow for a high rate of movement. That's going to have some serious implications on the way that the channel actually works. So large ion currents result in high rates of change. Then you also have, of course, the electrical chemical gradient that we discussed already, right? Especially established by carrier proteins like the sodium potassium ATPase. So you have to have a gradient for a channel to actually work. And then also channels are going to be selective to not only certain ions, but they're also going to be selective to the charges of ions. So for example, a positively charged ion is only really going to move through a negatively charged channel. And the opposite is true. So for example, a chloride ion channel is going to be positively charged. So basically you can attract the negatively charged chloride ions. And then you're going to see that voltage dependent channels, they're going to change their conformation with response to the membrane potential when you have depolarization, for example. So here's the patch clamp method, and there's a couple different techniques that are discussed over here. So you have just the regular cell attached recording where you apply a little bit of suction. So you have this little tiny cannula. It's really, really small. And that cannula is so tiny that you're able to isolate one single ion channel. So pretty amazing stuff. So with this first method, you're basically just applying a little bit of suction, so you're preventing any movement of ions around the environment. You're only going to allow for movement through the ion channel. Then you also have the inside-out recording, so that you actually remove the channel from the cell, and so you're able to see, you're able to manipulate, currents in different ways. There's also the whole cell recording, so that's going to actually involve the entire. You can actually see what's going on within the cell. You basically remove an entire chunk out of the cell wall, the cell membrane. And then you have the outside out recording. And so you're basically able to flip the direction of the channel. So what used to be on the inside of the cell is now going to be on the inside of the cannula. So let's watch a little video that talks about these different methods. And again, this is like the way that you're able to actually measure single ion movement across a channel.
The patch clamp method is used to study the properties of a small patch of membrane. In this technique, a glass pipette with a very small opening is used to make tight contact with a tiny area or patch of neuronal membrane. After the application of a small amount of suction to the back of the pipette, the seal between pipette and membrane becomes so tight that no ions can flow between the pipette and the membrane. Thus, all the ions that flow when a single ion channel opens must flow into the pipette. The resulting electrical current, though small, can be measured with an ultra-sensitive electronic amplifier connected to the pipette. This arrangement is the cell-attached patch clamp recording method. A record of the current flowing through a single ion channel reveals when the channel is in an open or closed state. The currents flowing through single channels are called microscopic currents, to distinguish them from the macroscopic currents flowing through a large number of channels, distributed over a much more extensive region of surface membrane. The technique can be used to compare the response characteristics of different channels. For example, depolarizing voltage pulses applied to a patch of membrane containing a single sodium channel result in brief currents, the downward deflections. The sum of many such current records show that most channels open in the initial 1 to 2 milliseconds following depolarization of the membrane. The process by which sodium channels close is called inactivation. When the membrane potential is depolarized, voltage sensors, indicated by the plus symbol, allow the channel gates to open and current flows inward. After less than 1 millisecond, the channel inactivates when an inactivation gate swings up and blocks the pore. The pore remains blocked even during prolonged depolarization. The pore then closes and the inactivation gate opens as the membrane potential returns to resting levels. A macroscopic current measured from another axon shows the close correlation between the time courses of microscopic and macroscopic sodium currents, reflecting the opening, closing, and inactivating of channels. Most channels open in the initial 1 to 2 milliseconds following depolarization of the membrane, after which the probability of channel openings diminishes because of channel inactivation. Technical modifications have enhanced the patch clamp technique. For example, if the membrane patch within the pipette is disrupted by briefly applying strong suction, the interior of the pipette becomes continuous with the cell cytoplasm. This arrangement allows measurements of electrical potentials and currents from the entire cell and is therefore called whole cell recording. Two other variants of the patch clamp method originate from the finding that once a tight seal is formed between the membrane and the glass pipette, small pieces of membrane can be pulled away from the cell without disrupting the seal. Simply retracting a pipette that is in the cell-attached configuration causes a small vesicle of membrane to remain attached to the pipette. By exposing the tip of the pipette to air, the vesicle opens to yield a small patch of membrane with its former intracellular surface exposed. This arrangement, called the inside-out patch recording configuration, allows the measurement of single-channel currents with the added benefit of making it possible to change the medium to which the intracellular surface of the membrane is exposed. Alternatively, if the pipette is retracted while it's in the whole cell configuration, a membrane patch is produced that has its extracellular surface exposed. This arrangement, called the outside-out recording configuration, is optimal for studying how channel activity is influenced by extracellular chemical signals, such as neurotransmitters. This range of possible configurations makes the patch clamp method an unusually versatile technique for studies of ion channel function.
All right. Cool. So with the patch clamp method, you're basically able to look at those microscopic currents. Microscopic currents due to the flow through one single individual channel. And so you can measure specific ions flowing through specific membrane proteins. Macroscopic currents, those are going to be representing more large surface area. So patch clamp method is looking for more of those microscopic type currents. And so here are a couple different examples of patch clamp methods that could be either used to look at like sodium channels versus potassium channels. So here's being able to isolate the sodium channel, you have to wipe out the potassium channels. And so for this specific experiment, they used cesium, which is on the periodic table, right? Cesium is one of our elements, and cesium actually blocks those potassium channels. And so that's the mechanism of action for that. And then you're able to just look at specifically sodium channels and how they operate. So as that narrator in the video was discussing, how you have a downward deflection once you depolarize the membrane, and then you have a brief movement of sodium, which is going to be rapid and transient, right? It's going to be rapid and transient, meaning that even if you keep on applying that depolarized charge, eventually those voltage-gated sodium channels are going to close up and they become inactivated. That's going to be opposite of potassium, though. Potassium, those voltage-gated channels are going to remain open. So voltage-gated sodium channels stay open for a brief period of time, and then they inactivate much more rapidly. Yes?
So the purpose of adding cesium to block the potassium, is it just to study sodium?
Specifically looking at the sodium channel.
But if you wanted to look at potassium,
then you could block the sodium channel. So you could use tetrodotoxin, for example, to wipe out the sodium channel. Or you could use any sort of sodium channel blocker. You could put like lidocaine or Novocaine, benzocaine, any of those types of drugs, and you can wipe out the sodium channel so you can specifically look at the potassium channel. Yeah, so if you want to look at one ion, you're going to have to wipe out the other channels so that you can just look at that one ion channel. So that's a really great question. So here's the opposite, right? So for this experiment, they used tetrodotoxin. to basically disable those sodium channels, right? Tetrodotoxin's a sodium channel blocker. And so now you can specifically look at what potassium does. And again, potassium is going to do basically the opposite of what sodium does. You're going to have not only an upward deflection when you have depolarization, so you're going to get that change in direction of movement of the ions, and you're also going to have a delay in the opening of those channels, right? Versus sodium, it's rapid, right? It happens like basically right away. Potassium has a little bit of a delay, and then those channels remain open and they do not deactivate as you continue the depolarization. So in summary, these channels are going to be voltage gated, right? They have to be exposed to depolarization in order for them to open up, but they're going to open up at different times. Potassium takes longer to open. Sodium channels take a short period of time, right? 1 to 2 milliseconds. It's going to be much more rapid. And again, they have voltage sensors on the proteins, right? So in other words, when there's a current that's moving through the membrane, then you're going to have those proteins getting shifted depending on what charge the proteins are. So you have a positive charge moving through the cell membrane. If the proteins are also positively charged, there's going to be a movement in the conformation of the shape of the ion channel. And again, here's your sodium channels versus potassium channels opening and closing. So with right as soon as you have the depolarization membrane potential, then the sodium channels open up right away, transiently, because then they become inactivated, even with prolonged depolarization. But with prolonged depolarization, you're going to have those potassium channels opening up a little bit slower, but then they remain open as you have the depolarization running through. So sodium channels are quick to open, potassium channels are slow to open, but they stay open longer. So these are different types of toxins. We already covered a couple of them, like we talked about, for example, tetrodotoxin, which operates very similar to other sodium channel blockers like lidocaine, benzocaine, cocaine, any of those sodium channel blockers. Bless you. There's other compounds that also do similar effects. So saxitoxin, that's going to be associated with the red tide. So this is a picture over here of red tide. It literally looks red. So that's a red type of algae that basically flourishes during certain times of the year. And you want to avoid consuming anything that might have been contaminated with this type of algae because it can kill you. It basically operates as a sodium channel blocker, which can be extremely neurotoxic. It can paralyze people. So it's a paralytic type of contamination. There's also a cone snail that does something similar. So conotoxin does a very similar type of effect. It's a sodium channel blocker. Then you have different toxins like alpha toxin, beta toxin. They're both released, secreted by certain strands of Staphylococcus aureus, as well as scorpions. And there's other animals too that can secrete these types of toxins. But what they do, they affect those action potentials. For example, they can prolong the action potential. They can also cause depolarization of, they can cause opening of sodium channels at lower thresholds. So they lower the threshold of opening up. And then there's ones that affect specifically potassium channels. So dendrotoxin is one that is talked about quite a bit in your textbook, as well as these other two over here, apamine. I don't even know how to pronounce this one. Charidotoxin, I guess. Anyways, so lots of different types of toxins. The main ones that you're going to be responsible for are like tetrodotoxin. Let's watch a little video on red tide because it's a really interesting phenomenon. I mean, I'm sure all of you guys, especially seafood enthusiasts, probably heard about red tide. You want to avoid eating contaminated fish.
Red tide is actually just an algae bloom. However, it can carry with it biotoxins and create sicknesses such as PSP.
During certain portions of the year, then that level of that biotoxin can get high enough that it can cause paralytic shellfish poisoning. The red tide term is because at some point there may be enough of that algae in the water to turn the water slightly red, but the majority of the time there is actually no change in the color of the water. Mild symptoms would typically include numbness or tingling of the tongue or lips. It can progress to numbness and tingling of the fingers and toes, to paralysis of the arms and legs, and then as the level grows even higher, patients can develop difficulty breathing due to paralysis of the respiratory muscles. Since these symptoms can develop rapidly towards respiratory failure, it's better to be safe than sorry and to go right to the ER where there is emergency medical staff, including mechanical ventilators that can keep people breathing even if they stop breathing on their own.
These beaches, these areas have never really shown to have intertidal red tide. And so now that ocean conditions are slightly changing and we're moving into a new normal, we've observed toxins. We've had to close beaches since January of 2018, and we're being diligent to sample and work with the co-managers to make sure the beaches are ready and clean and people can go back to harvesting.
Oh, you're driving?
Yeah.
So where are we headed right now? Camino State Park. Camino Island. These are good. Pretty good size. Decent. A little small still, but we collect about 15 butter clams, bring them to the lab.
Every species of clam, mussel, oyster takes up and releases the naturally occurring toxins at different rates. The butter clams are notorious for holding on to toxins longer than almost every other clam.
In this area, me and Mike worked here for almost 20 years, and we've never had PSP problems. Paralytic shellfish poisoning, where it's like a red tide in the clams up in Saratoga area. And it showed up last year, so we're just trying to make sure the beach is clean and safe for tribal harvest. There you go. We got one sample ready to go to the lab. Throw it on ice.
The problem with the biotoxins is cooking isn't going to get rid of those.
Freezing does not destroy this toxin. The toxin can neither be smelled nor tasted. So if you are harvesting clams or other shellfish, there's no way that anybody's going to be able to tell just by looking at it or smelling it.
Camino Head, Island County, species we turned in butter clam. PSP result 66, has to be below 80 to pass. Parts per thousand, 80. NTD means no trace detected.
These toxins, they're naturally occurring, so they've always been around throughout history. They're not going to go anywhere. There's no way to eradicate them, nor do we want to. Everything has its place in nature. Everything has its place in our environment. So they're there, and it's just our responsibility to work with them. Be responsible if you plan on clamming or going out for shellfish. Just to give a look on the website, maybe see which beaches are better.
So super interesting stuff. So that is red tide. Again, very similar to the way tetrodotoxin works, right? So saxitoxin is very similar. Sodium channel blockers. Yes.
So which ones are the ones you want us to know?
Tetrodotoxin is a good one to know. Maybe saxitoxin. I haven't determined whether I'm going to put that on an exam or not. These other ones, I'm not going to test you on these other ones. So there's a lot of stuff on here that I'm not going to hold you guys responsible for.
So those are like regular like clams that are eaten by people. So they just test like a batch or everything?
That's a great question. I mean, some people recommend to not eat certain types of shellfish during certain times of the year, just because those certain months have a higher propensity for red tide. I don't know them off the top of my head, but they'll even say like don't buy them at the grocery store during those times of year if it's like fresh seafood, right? Because it could potentially be toxic, right? You don't know that. You can't. Yeah, me too. Yeah, I'm a huge fan of seafood. Some of it can be a little sketchy, right? The world's largest tapeworm, by the way, is from raw fish. So Diphyllobothrum latum, that's the world's largest tapeworm, and it's found in raw fish. So for those of you who are fans of sushi like I am, keep that in mind. Maybe bring your friends out to a sushi date sometime and be like, hey, by the way, I learned from my professor, world's largest tapeworm comes from raw fish. You know, when the way sushi, the fish is usually treated, they usually freeze it. and that could potentially help to reduce the parasites. So, yeah, but sushi is uncooked for the most part. Yeah.
But people say that you should do parasitic cleanses because
I heard that too.
Wait, what's a parasitic cleanse?
it's like you have, I think it's like activated charcoal and
Oh, okay.
a certain pill that people have been taking or something. But you have to go on a specific diet because if you don't, then they cling to like certain stuff that you eat. So like plants and fruits.
Yeah. I mean, I think here in the West, we're like over sterile for the most part. For example, like autoimmune diseases. You don't really see autoimmune diseases as much in like developing nations where they have like lots of exposure to parasites. Whereas over here in the West, we're like super sterile. Like everything is always clean, disinfected, and then it makes your immune system kind of bored. And then all of a sudden your immune system starts to attack itself. That's why you see autoimmune diseases at higher rates in Western developed type nations. So anyways, I think maybe we should have more parasites in our diet. Anyway, so here are some other toxins. Well, more details about those toxins. So we talked about alpha toxin already. So this is from the scorpion. Again, I'm not going to try to pronounce that. So it's a scorpion toxin. But this one's going to affect sodium channels as well, and it's going to prolong them. They can also diminish the threshold at which the sodium channels open up. So they can actually depolarize at a lower membrane potential. And so that can have some serious effects in terms of like the way the neurons actually function. Here are some other things I'm not going to test you on, but these are different types of voltage gated ion channels and the genes that are associated with those ion channels. I am not going to test you on these. So they're mentioned in your textbook, but I'm not going to hold you responsible for that. Here are some other categories, right? So we talked about ligands before. So these are your ligand-gated channels, which work as a lock and key type mechanism. So neurotransmitters are going to be ligand-gated. Cyclic nucleotide gated, again, those are going to increase your second messengers, cyclic AMP, cyclic GMP, right? So that's why they're called cyclic nucleotide gated channels. And then you have your TRP channels. These are your transient receptor potential channels. And thermosensitive channels, mechanosensitive, those both fall under that category. And with thermosensitive, we're going to discuss again capsaicin and how that also operates very similar to the way heat operates. And so this is a couple different examples of how these channels might open up due to different types of stimuli. So over here you have calcium channels, how those operate. You have also glutamate type channels, so ones that operate from glutamate receptor activation. You also have channels that open up specifically due to changes in light, right? So those are especially going to be like your photoreceptors. So we're going to talk about that when we get into special senses with the way the eyes work and how the retina is going to respond to different changes in light frequencies. And then again, you have temperature receptors that are going to change to fluctuations in temperature. And then over here in your auditory system, those are going to be due to displacement of the hair cells, secondary to vibrations in the air, right? That's how you perceive sound. So those are just different examples of how ion channels may open up to different types of stimuli. And so here's your temperature sensitive receptors. So these are your transient receptor potential channels, so TRP, and there's a bunch of them. There's ones that are specifically geared towards cold. There are some that are going to be specifically geared towards detections of heat. And don't bog yourself down with remembering every single one of these different types of temperature channels and how, what degrees they respond to. But that's just an example of how this how these work on a spectrum, right? Different temperatures to different channels. So first, let's start talking a little bit about ion channel pores. And the ion channel pores are going to be large enough for only the one ion that they're specialized in to open up. So You have channels that are going to be specifically geared towards sodium, specifically potassium, or specifically chloride ions. They're all going to be shaped differently. They're going to have different charges on them, right? So they're going to be specified. So they have a selective filter. Again, the opening to these pores are going to have the opposite charge of the ion that they're meant to attract. So for a sodium channel, the opening to the pore is going to be negatively charged. So it can attract the opposite charge ion. Other things that are going to affect the selectivity, we just mentioned the diameter. There's also going to be things like the atomic radius. So these are like your naked type ions not associated with water. There's also ones that are going to be associated with water, so those are considered hydrated. And then again, the electronegativity of the ion is going to affect the ability for ions to move through different channels. So here's an example of your potassium channels. You got your potassium pore, the opening that's only going to allow for potassium to come in. You also have your sodium channels as well as your chloride channels. And these are all voltage gated, and they are going to be an opposite charge to the ions that they're meant to attract. The channels, the voltage gated channels are going to be a little bit larger. They have more of the transmembrane structures, and those transmembrane structures are what you find embedded within the membrane, and those are going to change in shape and conformation due to whether or not the membrane is being depolarized. And so when you do have depolarization, then you're going to have movement of those charged sensors away, right? So that's kind of what allows for those channels to open up. And then hyperpolarization is going to do the opposite. It's going to make those sensors move inward. And so here is an example of one of your voltage-gated channels. And so here's the transmembrane structure, the transmembrane proteins that are going to have a specific charge to them. And then once you have depolarization, then those charged proteins are going to move. They're going to actually change in their conformation. And once they do so, that's when those channels actually open up. And so depolarization is going to help open up those channels. And then if you have hyperpolarization, those channels get shut, closed again. So let's watch a video talking about the charged sodium channel.
You can see the voltage sensors colored in pink in this animation. Because the voltage sensors are positively charged, they're repelled by other positive charges. So at resting potential, which is around negative 70 millivolts for most neurons, there will be more positive sodium ions outside the cell than inside the cell. The voltage sensors move down, causing these purple gates to snap shut. So at resting potential, the gate is closed. Sodium ions cannot get through. This corresponds to the flat line at the beginning of our action potential graph. However, if the neuron is receiving an excitatory signal, the number of sodium ions increase within the cell. And if we reach the threshold potential, this will repel the positive charges on the voltage sensors and cause them to move up. This, in turn, causes the gates to open, and sodium floods into the cell. This is represented by the long, steep part of the graph, where the potential is rising very quickly. However, around positive 30 millivolts, the inactivation gate will engage. This will block the further influx of sodium, even though the voltage gate is open.
All right, cool. So that's a really nice little animation that shows how those channels actually change shape due to the changes in membrane potential. So here are some other different types of ion channels. So you have glutamate receptor type channel. So this one is going to have, for example, a clamshell type structure. To me, it looks more like a Pac-Man, but they call this a clamshell type structure. Once you have glutamate binding to the receptor site, then it's going to basically change those transmembrane domains. So you have your transmembrane proteins down here. These are the ones that, when you say transmembrane, it goes throughout the entire membrane, the phospholipid bilayer. And then when you have stimulation of those receptors, those transmembrane proteins move and they change their conformation. So here's a couple diagrams that depict how this all works. So you have glutamate receptor binding to that clamshell type receptor. And then upon binding, the transmembrane protein basically opens up. So you have opening right over here. So before it's bound to glutamate, it's going to remain closed. Once it binds to glutamate, then those channels are going to open up. And here is your cyclic nucleotide gate channels, right? So once the channels get stimulated, then you're going to have cyclic nucleotides coming into play to help opening up those channels. So this would be an example of something that you might see responding to changes in light. So here's one example. So you got the transmembrane proteins over here. You got your cyclic nucleotide binding proteins that are colored in green. And then once these get stimulated by changes in voltage, and including changes in light, for example, for like photoreceptors, then you're going to have those transmembrane domains changing conformation and opening up, allowing for ions to come through, moving through. Now let's talk about the heat channels that are also going to be responding to capsaicin. So these are your temperature-sensitive TRP channels, and they're very similar to your other voltage-gated channels, except these ones are going to be permeable to calcium. And so whenever these get stimulated, then calcium is going to be able to come rushing into the cell. And again, they respond to heat as well as to capsaicin. I'm going to show you a video in a second talking about how capsaicin can be used therapeutically. So TRPV1 is the one that's specifically geared towards heat, changes in heat. Again, it's going to be a calcium permeable channel. So calcium is going to be able to come through. And when you have any sort of changes in temperature, then it's going to effectively change the conformation of those proteins. And so those are going to actually be able to open up, allowing for calcium to come in. And here's how capsaicin has been used therapeutically. It can be used in multiple different ways. There's actually capsaicin patches that people can use. There's also capsaicin lotions or creams that people can use. This really hurts when it's being applied, right? I don't know if you guys ever had like really hot, spicy peppers on your skin, but when I was living in the West Indies, I like the first thing I did was I went to the farmer's market, bought a big bag of like locally grown peppers and stuff. And so I was like cutting them up at home and making my own like hot oils and whatnot. And for days, I was like, my fingers are burning. So you're supposed to use gloves when you're preparing peppers like that. And I was an idiot back then. I didn't know that you had to do that. And so like for days, like my fingers were just like burn like crazy. So this is really, really painful. So patients that do have like diabetic neuropathy, for example, they usually apply first lidocaine. And so they give the patient lidocaine first at the area, and then they apply the capsaicin patch. And what this does, when you have activation of these channels for a long time, for example, prolonged exposure to capsaicin, eventually that's going to basically numb those channels. It's not going to let those channels operate much longer. And so you basically overstimulate them for a short period of time, and then they're not going to be stimulated any further. And this can be used for diabetics, especially, right? Diabetics get neuropathy. The thing about diabetes is that when you think about like sugar, think of it as like little shards of glass floating around in your circulatory system. And when you have like too much glucose levels in your blood, it basically wreaks havoc on so many different parts of your body. It causes, for example, blindness. It can cause renal failure. A lot of your dialysis patients are going to be either diabetics or hypertensives. That's like the majority of those types of patients. And the same thing with your neurons. So hyperglycemia is going to cause damage to neurons. So let's watch a little video on capsaicin being used for neuropathic pain.
Chili peppers don't just put fireworks in your food. They also have medicinal properties. The active ingredient is the capsaicin, concentrated in the ribs and hip. That's what causes the burning sensation on your tongue or skin.
When we experience pain, the body undergoes a stress response. We'll feel warm and may even break into a sweat. These are clear signs that your metabolism is hard at work.
That warmth can also be used to ease tense muscles. It's often helpful for joint and back pain. Capsaicin patches and creams are especially popular.
Capsaicin dockt an einen bestimmten Rezeptor.
Capsaicin binds to a specific receptor in the cells of the body, a calcium channel. The capsaicin stimulates neurons that are sensitive to heat and warmth, which gives you that localized sensation of warmth.
Eine lokale Wärmereaktion entsteht.
Tina Meinke studies the effects of capsaicin. She wants to use it to help ease severe neuropathic pain, which is usually quite difficult to treat. The doctor works with high-dose patches that contain 40 times more capsaicin than the ones available over the counter.
Dementsprechend ist auch das Anwendungsgebiet.
These patches are prescribed for certain kinds of pain. The high concentration of capsaicin leads to a kind of defunctionalization of specific nerve fibers in the skin. Afterwards, it takes a while for them to regenerate.
The effect can last up to 3 months, which can help ease severe nerve pain. But both the high-dose patches as well as the non-prescriptive.
Whoops. That's not good. That's a bummer. Well, if that doesn't work, then just watch it on your own time for the rest of it. So it's a good video. I really like it. I mean, a huge fan of peppers in general. I mean, they help like increase your metabolic rate. Also, they can actually help to minimize anxiety. So if you're feeling like really anxious and you like put some like cayenne pepper on your food or whatever, that can actually help to minimize your anxiety. So really, really cool, the effects of capsaicin. So here's your mechanical receptors. So these are going to be part of the piezo family of receptors. And again, these are going to change their shape and conformation due to, changes in pressure. So they can respond to, for example, light touch. Let's see if this link for allodynia works, because allodynia is a neurological condition where anything is going to provoke extreme amounts of pain. All right, good. Looks like this one does work.
Allodynia is defined as pain from a stimulus that wouldn't normally provoke pain.
I don't know why that other video wasn't working.
People who experience nerve pain are very sensitive to touch. For example, someone with this condition could feel pain just by putting on a pair of pants. Allodynia is a common symptom of peripheral neuropathy that can significantly impact the quality of life for those affected. Types of allodynia. Thermal allodynia refers to when someone experiences pain from mild, hot, or cold temperatures. While someone may feel discomfort when it's freezing or too hot, someone with allodynia feels pain when there's a slight change in the temperature. Mechanical allodynia. This type of allodynia occurs when there's a slight movement across the skin, such as fabric touching skin. Tactile allodynia. This occurs when light pressure is applied to the skin and is also referred to as static allodynia. Brushing your hair doesn't usually cause pain. Someone with tactile allodynia would find it painful. Examples of allodynia. A burning or stinging sensation when brushing or combing hair, pain while wearing clothing that is too tight, has tags, stitching, or rough textures, pain from shaving one's face, pain with light touch, such as a gentle pat on the back or a hug, pain from the pressure of a seatbelt or a hug, pain from exposure to heat, such as taking a warm shower or being in a warm environment, sharp and intense pain from exposure to cold weather or cold water from a bath or shower, pain from a slight breeze or draft of air, pain from light pressure from a massage or acupuncture needle.
All right, I think we get the picture, right? So basically everything is painful when you have allodynia. Horrible. Really, really horrible condition to experience. Yes.
I know allodynia as a result of light touch or any stimulus, but how does this happen, like the molecular level?
So it could be part of like neuropathy. So it could be part of like diabetic neuropathy, for example. So when you get neuropathy, you can either get like pins and needles, right? So like that feeling of numbness, right? That's like paresthesia, but you could also get pain as well. So that's why when they're when I was showing that video of like the capsaicin patches that they use, that's another way to treat neuropathy, right? And so you basically overstimulate those receptors and it kind of wipes those receptors out. And so for allodynia, you could potentially use capsaicin to treat that as well. But there's other drugs too. Some people take gabapentin, pregabalin. So there's drugs that can help to reduce neuronal firing. So we're going to talk about GABA receptors later on. So there's different ways you can try to treat that, but yeah, horrible stuff. So these different receptors, these receptors are going to operate with different ions. So some are going to involve sodium, some involve potassium, some involve calcium as well as magnesium. And again, when you have any sort of change in the environment, like change in like the pressure to the affected area, those channels are going to open up. So tension builds up. The tension, when it builds up, opens up the levers, and then that's when the channels actually open up. So I'm going to skip playing this video over here because I want to get on with it. But mechanoreceptors, they're going to fall under the piezo channel family. And with these channels, they have 3 different units, and those 3 blades are the portions that are receptive to changes in the environment, right? Changes in shape. Now let's talk about gradients. So we're going to revisit some stuff that we've already kind of talked about already, glossed over. Yeah. So sodium, potassium, ATPase, again, is going to be one of the most important channel proteins in the body. That's going to be especially for like neurons, right? Maintaining that concentration gradient. You're also going to have calcium ATPase pumps that help to move calcium in different areas. Calcium, of course, is going to be really important for neurons to function as well. So you especially find that at the ends of the axon. So when you have depolarization signal being sent all the way down the axon, calcium is going to influx into the terminal of the axon and allow for example, neurotransmitter release. So those vesicles that hold neurotransmitters are going to be able to release. So you have to have the ability to maintain a calcium gradient as well. Other areas that I'm sure you guys remember from A&P, so you have the circa, which is part of your muscles, right? You find circa, which is like sarcoplasmic reticulum, in skeletal muscle. You also find it within your cardiac muscle as well. And basically it like removes all the calcium that's within the cytoplasm so that it can be released when you have stimulation of those myocytes. So here's your sodium, potassium, ATPase. You get an exchange of 3 sodium for 2 potassiums. I don't really care so much about the rates of change. For example, the rates of change of potassium versus the rates of change of sodium. That's not super high yield, but just know like 3 sodiums for 2 potassium. Here are a couple experiments that show if you manipulate the concentration of ions either inside or outside the cell, that's going to affect the ability for the sodium, potassium, ATPase to function properly. So over here, the experimenters removed potassium, and so that basically affected the ability for the channel to work, right? Because you have to have both of those ions present for these channels to actually operate properly. And then if you remove ATP, then those channels also don't work, right? ATP is going to be really important for allowing those channels to function properly. And then this is revisiting stuff that we already talked about, but just let's briefly go over it again. So you got your antiporters. So antiport means that you're seeing bidirectional movement, right? So you have one ion moving in one direction and then another ion moving in the opposite direction. So here's a sodium calcium antiport, sodium hydrogen antiport. Then you also have your co-transporters. So they need to have, they use an electrochemical gradient as the source of energy for the ions to be able to move. So you have to have an electrochemical gradient for these to function properly. These also can be antiports or co-transports. And here's a sodium-calcium exchanger. So with a sodium-calcium exchanger, you're going to be exchanging 3 sodiums for 1 single calcium ion. So here's how this one works. So you got 3 sodium being exchanged for every calcium being exchanged. And that basically summarizes your ion channels. You guys have any questions? Let's go through a handful of these questions, and I want to get into 6.1. So let's maybe do like 3 questions from those quizzes, and then we can move on to the next series. All right. All right, so this is a case of neuropathy, right? And with this patient, they're being treated with capsaicin. And the way that capsaicin works, it's going to operate by activating the TRPV1 receptor. And that's the same receptor that is going to be involved in heat sensation as well. So you prolong the activation of that receptor, and basically it renders that receptor unworkable, right, over time. And so that's why D was the correct answer. This is different than voltage-gated sodium channels, right? So like tetrodotoxin, lidocaine, those types of drugs, they work on voltage-gated sodium channels. They can, reduce pain sensation, right? That's what you use if you do like a nerve block, for example. The piezo ion channels, those are the ones that are going to be mechanoreceptors. So those change, like those are like light touch, for example. So whenever you're manipulating the skin, moving the skin, that's going to open up those channels, the piezoreceptors. Let's do a couple more. All right, good. Ion channels are involved in generation of action potentials, open or close in response to changes in the voltage or ligand binding. Good. All right, one more, and then we'll continue on with 6.1. Okay, proteins that establish ionic gradients are called active transporters. They're not voltage-gated ion channels. The ones that establish the actual gradient, the concentration gradient. Okay, does that make sense? So to establish the concentration gradient, you have to have energy involved. So you have to have energy in the form of ATP. The greatest example of that is going to be your sodium-potassium ATPase. Right? That helps to establish that concentration gradient. Without that concentration gradient operating, then you're not going to get a depolarization signal at all, right? Because ions are going to eventually, you're going to get, what do you call it? Equilibrium, right? You don't want equilibrium in a neuron. Otherwise, the neuron's not going to fire properly. So you need to have active transporters. Yeah, let's do one more. Are we clear on this one? Because the majority of you guys got this one wrong. Yeah? Okay. One more. And then we'll move on. All right, so that's the patch clamp technique. This one, they just applied a little bit of suction, just enough to basically grab hold of that one single ion channel. And so with that, it's still attached to the cell. Right? So this is not a whole cell patch clamp. If you did the whole cell, that means that you removed that portion of the cell membrane, and then now you're able to actually measure what's going on within the entire cell. Whereas with this technique, where you're just isolating that one single individual ion channel, that's going to just be one single ion channel, and it's still attached. So that's why it's called the cell-attached patch clamp. Measuring the flow just through one single channel. So do the rest of these questions on your own time. And speaking of quizzes questions, because you guys have your next upcoming assignment, or your pre-topic assignment due on Sunday, do you guys still have access to the textbook? Yeah. You did? You went in there today? And, yeah, that's good. Okay. So it says that, but it's not true, right? It says it's going to be available until. Huh. Okay, good. I'm glad that that's still working. So, in other words, for your pre-topic assignments, do what I originally intended you guys to do. So, go into your textbook, do your quiz questions at the end of each of those chapters. There's 5 for this section, so sorry. Again, not all the stuff that you're going to be quizzed on is completely relevant for this class. But, do those quizzes. I'm not grading you for accuracy. I'm mostly grading you for completion. So just do it.
Do you want us to submit a screenshot?
Yeah, just submit a screenshot. You can submit a screenshot for each one and maybe put it into a Word document. So I see that you did every single one of those end of chapter quizzes. So cool. Let's switch gears now. Let's talk about synapses and synaptic transmission. And so with synaptic transmission, it's the way that neurons are able to communicate with each other, or how neurons are able to communicate at the neuromuscular junction, right, to allow for flexion of muscle, for example. So we'll talk about that in a second. Here is a summary of all the stuff we're going to be discussing in this section. So we're going to talk about different types of synapses, whether they're electrical versus chemical. We're going to discuss some of the different structures that you find in the synapse that allow, for example, for synapses to adhere to each other, as well as for the movement of vesicles, because you have to have, depending on what kind of axon and synapse you're talking about, you're going to have different neurotransmitter release, and there's different structures within the cell that are going to allow for neurotransmitters to be released through exocytosis from a vesicle. We're going to talk about ionotropic versus metabotropic receptors. So ionotropic, those are ligand-gated versus G-coupled protein receptors, which will be your metabotropic. We're going to briefly discuss end plate potential. So there's the end plate potential EPP, and there's also the miniature end plate potential, or MEPP. We're going to discuss calcium because calcium is really important for neurotransmitter release. Also, we're going to discuss structures that are important for vesicles, how like the vesicles actually move. And then we're going to discuss some clinical conditions. So we're going to talk about myasthenia gravis, or even just like myasthenic conditions broadly. And then we're going to discuss also what Clostridium bacteria. So Clostridium is a species, is the genus of bacteria that is responsible for example, tetanus, as well as botulinum toxicity. So Clostridium tetani causes tetanus. Clostridium botulinum is what causes botulism toxicity, which is the most toxic compound known to humans, is botulism toxin. Doesn't take much of it to kill somebody. Yet we inject it in our faces all the time to, treat wrinkles, things like that. So fun stuff. And people in cosmetics make a lot of money off of Botox. That's a huge, huge source of income for all those like nurses that like to inject people. They call them injector nurses. So it's a fun field. That's what you should get into. You can make a lot of money doing that. You make more money than a neuroscientist, I'm sure. And what was your question?
It's not about this, but is it chapter 5, right?
Oh, the chapters that are part of the pre-topic assignments or assessment? Yes, it's 5. I think it's 5 through 7, as well as chapter 20-something. Yeah, 5 through 8, and 24 and 26. Don't do chapter 25. Out of all the textbooks in this class, that's the one that we're skipping entirely, is chapter 25. And and then I'm gonna have to like pick and choose, like do some hardcore cherry picking when we get towards cognition and cognitive neuroscience, because there's quite a few chapters in there that we're gonna have to kind of unfortunately gloss over. 6 quizzes. I'm sorry. Anyways, now let's discuss the difference between electrical versus chemical synapses. With electrical synapses, they're going to have gap junctions in them. So ions can basically move freely from cell to cell. It's very similar to what you see in cardiac myocytes, right? When you have depolarization of a heart cell, right, you're going to have gap junctions. You're going to have intercalated discs. That's where you have the gap junctions. The cells are attached through desmosomes. And then when one cell depolarizes, calcium freely flows from one cell to the next. And that's why in cardiac myocytes, you get synchronicity of the beat. When the ventricles depolarize, they all contract basically simultaneously. So it's very, very rapid. You also see bidirectional flow of ions. So you can get ions flowing in either direction. Versus chemical synapse, this is going to involve calcium, right? Once you get the depolarization signal sent all the way down to the axon terminal, then calcium is going to come in, and calcium is going to basically allow for the vesicles that contain neurotransmitters to release the neurotransmitters into the synapse. This is going to be unidirectional. So whereas electrical synapses move in both directions, chemical synapses are in one direction, and it's going to be a lot slower, relatively speaking, compared to an electrical synapse. It's still pretty fast, but relative to electrical synapse, much slower. So here are a couple images from your textbook that basically compare and contrast those electrical synapse versus chemical synapse. Electrical synapse, again, you have gap junctions that connect both of those cells together. You also have things like connexons, and connexons are going to be proteins that create those gap junctions. So you have a bunch of those different, subunits of connexons making the entire gap junction. Versus the chemical synapse, where you have calcium influx, which allows for release of neurotransmitters. So those little vesicles, they basically stay within the axon terminal until the axon gets stimulated. And then when it gets stimulated, then it can also allow for the release of those vesicles. So I included some images here that were kind of cool. These are like transmission electron microscope images that show the very end of axon. So this is the actual synapse right here between the receiving cell and the cell that's sending out neurotransmitters. And all those little tiny little circles over there are all those vesicles filled with neurotransmitters. So super cool stuff. Now with electrical synapses, because they're in direct contact with each other with gap junctions, they're going to have a smaller synapse. It's going to be very close, so it's going to be narrower rather. Not so much smaller, but it's going to be narrower. And then connexons. Again, connexons are what make up those channels, those little gap junctions. And those are going to be really, really fast. So you get basically instantaneous communication from cell to cell. And so here's a couple images that basically show you what those gap junctions look like. So here are all the different little connexins that make up the actual gap junction. So you have all those little subunits coming together to allow for basically a pore to be formed into the membrane. Connexons are going to be the little subunits. So if you have like a bunch of connexons all put together, then they eventually form the pore. Right? So they're going to be subunits of the gap junction. And you're not really going to find gap junctions as much as you find chemical synapses. Sorry, electrical synapses versus chemical synapses. Electrical synapses in the central nervous system are mostly going to be associated with the hippocampus. In other areas of the brain, though, you're going to see more of your chemical type synapse. So here's your hippocampal neurons, or interneurons rather. And again, that's one of the few places where you find these. And it allows for the synchronicity of neuronal firing. Right? So when one neuron fires, that signal can be sent really rapidly from cell to cell. So you basically get synchronicity of the entire structure of the hippocampus. Why is it called the hippocampus? Because it actually comes from Greek for sea monster. So it kind of looks like a little seahorse. And so that's actually what hippocampus stands for. And the hippocampus is going to be where you have storage of long-term memory. Right? So all of the long-term memory that you have, that's hippocampus. There's other areas in the brain that are going to be associated with like memory retrieval, as well as the formation of memory. You got areas of the temporal lobe. You got areas of the insula. You got areas of the prefrontal cortex that are all going to be involved in memory retrieval as well as memory storage. But when you have hardwired long-term memory, that's going to be within the hippocampus. So here's an image of what's going on in those cells. So you have your presynaptic neuron, and then you have your postsynaptic neuron. Presynaptic neuron, presynapse, that's going to be the sender. The receiver is the postsynaptic neuron. And when the presynaptic neuron fires, right away, you're seeing postsynaptic neuron being stimulated. So it's almost like instantaneous. Right? So there's a very little tiny lag from neuron to neuron, but it's almost instantaneous. So when you have one neuron firing, the next neuron is going to basically be stimulated and fire as well. Yes?
What is the result? I see all of them are star except one.
Yeah, it's sometimes you get some spontaneous type firing, but when you have a one-to-one, it's mostly going to be a one-to-one. You're going to have, whenever the presynaptic neuron fires, the postsynaptic neuron is also going to be stimulated and fire as well. So I don't know if that's going to be random firing or maybe it's receiving information from another neuron. Maybe it's like a different neuron that's stimulating as well. But whenever this one over here is getting stimulated, then you're also seeing the subsequent neuron being stimulated as well, like right away. Presynaptic neurons are going to be the senders. Postsynaptic neuron is going to be the receiver. And again, that's going to allow for the hippocampus to basically fire in unison. So it allows for synchronicity of neuronal stimulation. So here's your chemical synapses. Again, chemical synapses are going to be the ones that you find more often, right? And they use neurotransmitters. And we're going to discuss all those neurotransmitters next time. We're going to talk about like GABA, for example. We're going to talk about serotonin. We're going to talk about dopamine. We're going to talk about opioids. So for this one over here, you got your opioid receptor that's going to be responding to, for example, morphine, but it could respond to fentanyl, it could respond to heroin, it could respond to any of your opioid type compounds. And when you have stimulation of those receptors, then you're going to have an influx of calcium, and the calcium influx is what allows for the vesicles to release their neurotransmitters into the synaptic cleft. So, in other words, you first need the neurotransmitter, and you also need calcium. Otherwise, you won't get any sort of release of neurotransmitters. So you have electrical transmission. That's going to be the signal sent. You also have chemical transmission, and then you eventually have resumption of the electrical signal. And again, you need to have calcium movement to allow for these to work properly. And there's different types, right? The big categories are going to be inotropic versus metabotropic. So inotropic, that's ligand gated. Metabotropic is going to be G protein coupled type receptor. So here are the types of structures that you need for these neurons to operate properly. You need to have different types of filaments. So you have like these filaments that allow for the vesicles to be activated and to allow for the vesicles to move properly. You're also going to have different pools of vesicles, and they're going to be found within like the active site of the synapse. And so they basically are the ones that are most ready for exocytosis. So you have release of the neurotransmitters, for example. And then you also have, in the postsynaptic neuron, you have to have these little postsynaptic densities. And that's basically just a bunch of proteins that allow for anchoring. So you have anchors, and then you also have these postsynaptic densities being involved in plasticity. So we're going to talk about neuroplasticity later on. But when neurons, for example, if you're learning something, neurons are constantly like making new connections, making new synaptic connections. That's the whole purpose of synaptic plasticity, right? The brain is ever changing. So you get that with learning new things. You also get that with memory consolidation. And then that can, of course, be affected by conditions like disease, right? If you have, for example, Alzheimer's disease, Parkinson's, any of your neurodegenerative disorders that can affect neuroplasticity. So here are some of the structures that we just talked about. So here are your filaments, right? Those filaments basically guide your vesicles to move in different directions. They also help to, guide the synapse formation, so you can get plasticity in synaptic formation. So you have your synaptic vesicles. Those are going to be all those little tiny circles. Those are all filled with neurotransmitters. And then you have these, they call it dense projections. That's within the active area. Those are the active zones of the synapse. And yeah. Then calcium. Again, calcium is going to be really important for neurotransmitters to be released. So you have, again, electrical transmission. Then you have chemical transmission. So first you need to have a signal being sent down the presynaptic axon. Then you have chemicals being involved, right? So you have, for example, neurotransmitters being released. And then eventually you're going to have resumption, which means that eventually it's going to go back to normal, right? How does that happen? When you have neurotransmitters being released, you also get recycling of components of the neurotransmitters, right? You have reuptake of neurotransmitters. So, for example, with serotonin. Serotonin, when it gets released, it doesn't stay in the synaptic cleft forever. Eventually, it gets taken back into the back into the axon. Certain drugs prevent the reuptake of neurotransmitters. So, for example, cocaine. How does that work? It prevents dopamine from being reuptaked. So now dopamine stays within the synapse for a longer period of time. SSRIs, that's how those also work. They prevent serotonin from being taken back up into the axon. And so if you prevent that from happening, then you're going to have more stimulation of the receptors in the postsynaptic terminal. And again, calcium is also going to be really important in this whole process too. Yes?
I forgot. What did you say helps guide synapse formation?
The filaments. So filaments are going to help direct vesicles. You're going to find filaments in both the presynaptic terminal as well as in the postsynaptic terminal. So the filaments help to guide the direction of vesicles. So here's your end plate potentials. End plate potentials are going to be referred to as EPPs. And there's also going to be ones that are more spontaneous. And those are going to be associated with your miniature end plate potentials, or MEPPs. And for your spontaneous firing, sometimes you can get spontaneous firing due to acetylcholine being released. And acetylcholine, each of those acetylcholine molecules are going to be associated with an MEPP. So that's a mini end plate potential. And again, mini end plate potential, if you get more and more and more of those through like accumulation, you're eventually going to reach threshold, and that's going to allow for actual depolarization of the receiving cell. In other words, the postsynaptic cell. So here's a way that you can see the effects of depolarization of the neuron and how it affects, for example, a muscle, right? Because remember, with the way that muscles work is that you have to have release of acetylcholine that binds to nicotinic receptors in the muscle. And so you can record the postsynaptic potential in the muscle, as an example, and see how that relates to the stimulation of the actual motor neuron. So when you stimulate the motor neuron releases acetylcholine, and the acetylcholine is what binds to the nicotinic receptor on the myocyte, allowing for the myocyte to be stimulated and depolarize, and then eventually allow for contraction of the myocyte. And so if you use the calcium channel blocker, because remember, calcium is going to be really important in this whole process. If you administer a calcium channel blocker, that basically prevents any sort of stimulation from taking place. And so in other words, the postsynaptic membrane potential is going to remain dormant, right? It's going to remain at resting membrane potential. So you need to have calcium coming into play to allow for this whole process to take place. So again, if you use cadmium, which is your element from the periodic table, you administer cadmium, is going to block calcium channels, and that is the effect. When you block the calcium channels, basically you're not going to get any stimulation in the postsynaptic terminal. So here's an example. They use these dyes that allow you to track the movement of calcium. So here's a really short video that shows what this looks like. So if the internet wants to work. Is the internet here being finicky or what?
When is it not?
I know, right? It's often finicky. There we go. So right there is where calcium, I'm staying in the way. So all the colored, they used fluorescent dyes to be able to track the movement of calcium in this experiment. So like I said, that was a very short video, basically showing what's going on over here. So calcium-dependent fluorescent dye. That's what they use to be able to track the movement of calcium. And then if they give a calcium chelator, what does a chelator do? A chelator basically sequesters a substance. So there's different types of chelators. Like you can use iron chelators, you can use copper chelators. For this case, if you use a calcium chelator, it removes all the calcium, and then you don't get the effect, right? So you can get presynaptic depolarization, but then you're not going to get any postsynaptic effect whatsoever. In terms of neurotransmitter release and quantifying it. So you have the number of quanta released positively correlates with the number of vesicles fusing. So it's basically 1-to-1. Like the more vesicles you get released, the more effect you see in the postsynaptic terminal. So it's effectively a 1-to-1 relationship. So the more vesicles are released, the more of effect you get in the postsynaptic cell. So here's another image with electron micrograph. So this is using transmission electron microscopy. So with transmission electron microscopy, you can see right through the cell. All right, this is different than scanning electron microscopes. With a scanning electron microscope, all you can do is to see the surface of a cell. So with transmission electron microscope, you can see all those little tiny vesicles. Right? And then this over here, when you're closer to the actual synapse, this is considered the active zone. So whenever you actually get depolarization of the neuron, the active zone is what allows for vesicular release of those neurotransmitters. So in this image over here, you can actually see where those vesicles basically fuse to the wall of the cell, right? And so that's how you get exocytosis of those neurotransmitters. It's just through fusion. So the fusion also is going to be temporary. You get temporary fusion, and then you get the release of the neurotransmitters. The vesicles are going to be it's going to be energy dependent to produce these vesicles, right? So they're loaded with neurotransmitters. You have to have ATP to be able to allow for this process to take place. And they basically get stored in these vesicles until the vesicles fuse with the membrane during depolarization. So here is a little study that they did using horseradish peroxidase, which is basically a way to visualize what's going on within the cell. And so they're able to see all the different aspects of vesicular formation as well as the release of the neurotransmitters within the vesicles. And so they were stimulating endocytosis. So you have uptake of the horseradish peroxidase. Then you see endosomes. So the endosomes are going to be what allow for the production of these vesicles. And then you have the vesicles being filled with the horseradish peroxidase. And then you see how eventually the horseradish peroxidase gets released when neuron gets stimulated. So that was just one example of the study. So you see how the vesicles are produced. In terms of the things that affect the vesicles and how they're trafficked and how they're produced and how they're stimulated, there's a ton of proteins that are going to be involved in this, and I am not going to hold you guys responsible for all of them because there's a bunch. But this is a vesicle over here in this image, and these are all the different proteins. This is like 70% of the proteins that are involved. There's actually more that go beyond this. But you have things like snaps and snares. The snaps are going to be basically what allow for like the stimulation of the snares. Right? The snares are going to be the actual receptors. And there's different types of snares. So these are snap receptors. So you got synaptobrevin, syntaxin, etc. Like I said, I'm not going to hold you guys responsible for all of those. Just know the broad categories. So no snaps, no snares. Snares are going to be the receptors. And then NSFs, your textbook refers to them as NSFs exclusively. So those actually stand for N-ethyl malodamide sensitive fusion proteins. So we'll refer to them as NSFs for short. And SNAPs are going to be soluble NSF attachment proteins. And then you have snares, which are the actual receptors. So again, these are all the different things that these are involved in, right? These different proteins are involved at every step of the way, whether it is the creation of the vesicle within the endosome or the movement of the vesicle to the active zone and docking of the vesicle to the presynaptic terminal, and then the actual release of the neurotransmitter, and then the reassembling of the vesicle. So like I said, I am not going to hold you guys accountable for all these proteins because they do lots and lots of different things. So it's way beyond the scope of this class. But good to know. just the broad categories. So like your snares, so you have your vesicular snares. You also have the targets. So you have V snares and T snares. And they're also going to be involved, they're also going to require calcium to properly function, right? So if you don't have calcium, these vesicles are not going to properly function. And then eventually, ultimately, the goal is to allow the vesicle to attach itself to the presynaptic terminal so that it can release the contents of the vesicle into the synapse. So here are some other images from your textbook that discuss what these vesicles look like, all the different proteins are involved. Synaptobrevin is going to be listed over here as one of the blue little helical structure here. Then you have syntaxin, synaptotagmin, so a bunch of different proteins involved in vesicle function. Definitely know that calcium is going to be involved in this whole process, though. So that's really, really important. So now let's talk about some clinical conditions. So you have myasthenic syndromes. So with a myasthenic syndrome, you basically have dysfunction of neuromuscular synapses. So they can affect acetylcholinesterase. They could affect the cholinergic receptor itself. They could also affect the voltage-gated calcium channels. So if you affect any of these processes, you're going to get a myasthenic syndrome. And there's different types of myasthenic syndromes. There's Lambert-Eaton myasthenic syndrome. There's also congenital myasthenic syndrome. There's also myasthenia gravis, which we're going to talk about in a second. But basically what happens with these conditions is that you get muscle weakness. So with myasthenia gravis, that's going to be an autoimmune condition where you get destruction of the receptor, the cholinergic receptor, specifically the nicotinic receptor. And so with myasthenia gravis, if you destroy those nicotinic receptors, you're not going to get stimulation of the muscle. And so in other words, you're going to get muscle weakness. And how do these manifest? You can get general muscle weakness, but you can also get ptosis, which means that the eyes start to droop a little bit. So you can get ptosis of the eyelids. Let's watch a couple of videos on these. So here's Lambert-Eaton's myasthenia gravis.
Lambert-Eaton myasthenic syndrome is a neuromuscular junction disorder that was first described by Lambert, Eaton, and Rook in the 1950s. It is a disorder of voltage-gated calcium channels in the presynaptic motor neurons, the neurons that supply the skeletal muscles. The voltage-gated calcium channels do not allow calcium influx, causing reduced acetylcholine release into the synaptic cleft, and thus reduced muscle contraction and weakness. Lambert-Eaton myasthenic syndrome causes muscle weakness, which interestingly improves with activity, unlike the other neuromuscular junction disorder, myasthenia gravis, which manifests as fatigability. The more you use, the weaker the muscle gets. Myasthenia can be divided into ocular myasthenia and generalized myasthenia. Lambert-Eaton syndrome is a paraneoplastic syndrome because of its association with small cell lung cancer. Paraneoplastic syndrome can be described as an immunological phenomenon, but really are symptoms that occur at sites distant to where the tumor is. The presynaptic neuron houses vesicles containing acetylcholine at its terminal. When an action potential travels along the neuron, the voltage-gated sodium channels open, allowing influx of sodium ions inside the cell. This causes a positive electrical gradient, which then triggers voltage-gated calcium channels at the terminals to open up, allowing calcium ions to come inside the cell. Calcium ions trigger the vesicles to release acetylcholine, a neurotransmitter, into the synaptic cleft, into the neuromuscular junction. Acetylcholine binds to ligand-gated sodium channels, allowing sodium influx into the muscle, which eventually causes the muscle contraction. And because of this, when you check reflexes normally, you have reflexes present. The causes of Lambert-Eaton myasthenic syndrome can be either paraneoplastic or non-paraneoplastic. The thought is that the immune system mounts a response to voltage-gated calcium channels from cancer cells, creating antibodies against the voltage-gated calcium channels, which has a predilection to specific types of channels found in the neurons. Non-paraneoplastic causes are associated with autoimmune diseases such as Graves' disease and type 1 diabetes mellitus. In Lambert-Eaton myasthenic syndrome, these antibodies that are produced bind to the voltage-gated calcium channels at the terminal bulbs of motor neurons. They are found to bind to the P-Q type voltage-gated calcium channels, thus really preventing calcium influx and then subsequently the release of acetylcholine into the synaptic cleft.
So you guys get the basic idea there. So in other words, if you can't stimulate the release of acetylcholine, your muscles are not going to be properly stimulated. So that's Lambert-Eaton myasthenic syndrome, again, affecting the calcium channels. Then you also have congenital myasthenic syndrome as well, which is going to affect acetylcholine release. Did you have a question before we continue?
Yeah. So what is the difference between Lambert-Eaton and congenital? Because they both have to do with no calcium influx. You can't release acetylcholine. So are they just two different branches of this?
Basically, two different types of myasthenic type condition.
So what's the difference?
That's a really good question. I don't really know what the major differences are going to be in terms of symptoms. The major difference between Lambert-Eaton syndrome versus myasthenia gravis, though, is the fact that this one gets a little bit better over with repeated muscle use versus myasthenia gravis, which you get progressive worsening of symptoms as you use the muscles. And so you're fine in the morning, for example, and then throughout the course of the day, you start losing the ability to properly function, right? And also different mechanism of the pathophysiology, right? The pathophysiology in Lambert-Eaton, calcium channels are going to be blocked. Versus myasthenia gravis, it's a, again, autoimmune disease, but it's affecting the nicotinic receptors. So you're destroying the receptor, so acetylcholine can't do anything.
So the more you use it, the less likely it's going to actually do the movement?
So Lambert-Eaton is going to be the more you use it, the better it gets. Myasthenia gravis, the more you use it, the worse it gets. So here's a little video on congenital myasthenic syndrome.
Or CMS causes muscle weakness and fatigue. It is often difficult to diagnose and tough to treat. But as Tiffany Huertas reports, researchers are testing a new treatment designed to give patients their lives back.
37-year-old Elaine Whitehead steers her two sons toward after-school snacks and homework. It's a relief now to keep up with their daily routines.
Why would I not want to take care of my kids? And I couldn't.
Just 2 years ago, simple things like cleaning up after a meal or getting dressed were a challenge.
I started to have to brush my hair with my head over the side of the bed because I couldn't lift my arms up to do it.
Elaine saw multiple doctors, and nobody could tell her what was wrong until she met Dr. Stanley Ayadre. The neuromuscular expert says the symptoms of CMS are subtle and very hard to pick up.
What makes this different is in the setting of fatigue, they may also have eyelid drooping.
The reason he noticed it first was I have this lazy eye, and I've had it my whole life.
Doctors prescribe medication which helps some of Elaine's symptoms, but now researchers are testing a new drug for CMS, amifenamine phosphate.
We expect their eyelid droop to become completely normal, their speech to become completely normal, their swallowing difficulties go away, and the fatigue gets better.
After years with few treatment options. Tiffany Huertas, KSAT 12 News.
No more. Sorry about that. All right, so here is myasthenia gravis. Again, this is going to be a destruction of the receptors. So myasthenia gravis targets the acetylcholine receptors, nicotinic receptors at the neuromuscular junction. And again, they get progressive fatigability. It gets worse and worse and worse throughout the course of the day. For whatever reason, it happens more to females. And when I say for whatever reason, generally speaking, autoimmune conditions usually happen to women more often. Why does that happen? The thoughts are that women have more robust immune systems in general. So because of a more robust immune system, women are going to be more prone to making antibodies that start attacking themselves. Again, as I mentioned with autoimmune conditions, there's like an entire litany of autoimmune conditions. But you find these types of diseases in higher frequency in areas like the United States, Western developed nations that have like really like good sanitation for the most part. Your body basically gets bored, right? It's not fending off pathogens all the time. So it starts attacking itself basically. This could also be due to thymus issues. So if you have a thymoma or if you have thymus hyperplasia, the thymus gland is where your lymphocytes go to mature, like especially your T cell lymphocytes go to mature. This could be seen in women that are pregnant. It could be also postpartum after pregnancy. It could also be associated with anesthesia. So different things can bring this about. But notice on this image over here, you have weakness of the eye. So that's called ptosis. So when you have weakness or drooping of the eyelids, we call that ptosis. You can also get diplopia. Diplopia means double vision. So this doesn't just affect the eyelids, it also affects the extraocular muscles. So you're going to start getting cross-eyed, you're going to start getting double vision. It could also affect other muscles, like as the doctor in that video was mentioning, the muscles that are involved in swallowing. So they might have difficulty swallowing, they might have difficulty chewing. And again, it gets worse and worse throughout the duration of the day. So you get exertional fatigue, exertional weakness. It gets worsened with activity, but it does get better if you rest. So basically what happens is that you're allowing those receptors to basically, recharge themselves effectively. If it's really bad, you can get aspiration. And these patients could potentially get, like, for example, aspiration pneumonia, which, not a good situation. Yes?
If someone is giving birth, hypothetically speaking, the anesthesia isn't that strong, will that affect the birth child?
Will that affect the actual birth process? Oh, with myasthenia gravis, I want to say no, because with birth, you're dealing with smooth muscles, and that's going to be involving a little bit of a different process. So with myasthenia gravis, you're talking about the motor end plate. And so when you're talking about motor neuron disease, that means those are your voluntary muscles versus the muscles within the uterus, that's smooth muscle. And they're going to contract from different types of stimulus. So with smooth muscle contraction, especially during labor and delivery, that's going to be involving oxytocin allowing for contraction of the smooth muscles. So it's a different process. So that's that's a great question, but it doesn't have an effect. How do you test for myasthenia gravis? You can do an edrophonium test. It's called a tinselin test. And basically what you do is you administer the med and you see the effects being reversed. So let's watch a little video on this. This is actually pretty dramatic. So notice the ptosis on the patient's eye right here. All right. And as he uses his eye more, let me turn that down. That's like super obnoxious soundtrack. The more he uses his eye, the weaker and weaker the eyes get. Like notice he's starting to get diplopia, cross-eyed. So it basically completely alleviates the problem. So you can give drugs that are like acetylcholine esterase inhibitors. Acetylcholine esterase is basically what removes acetylcholine from the neuromuscular junction. And so if you give an acetylcholine esterase inhibitor, then that is going to basically allow for acetylcholine to linger longer within the neuromuscular junction. Again, there's no cure for this. Right? So this is permanent. You can give them different drugs. So you can give them, if it's like a thymus issue, you can remove the thymus, right? If they have like a thymus neoplasia or something like that. You can give them different types of meds, like pyridostigmine could also help in the treatment of myasthenia gravis. So again, the edrophonium, the way that works is an acetylcholine esterase inhibitor. It prevents the breakdown of acetylcholine. Cool. I think that's a good place for us to stop for the day. This is where we'll continue next time. We'll talk about Clostridium organisms, which are really exciting. Tetanus as well as botulinum. So cool stuff. Anyways, any questions? Rock and roll. So next time we'll continue here, and then we're also going to start talking about neurotransmitters, drugs, receptors, all that stuff.